Hi, my name is Julia, and today I'll be going over cell communication. So, cell communication is the way that our cells are able to talk to one another, and this comes in handy when cells need to coordinate their activities around the body and even inside the cells. And so, this applies to multicellular and unicellular organisms. So, before we start talking about cell communication in detail, we should go over some words that are involved and steps in the process. So first we have the secreting cell, which is this one right here, and this is the cell that will be sending out the chemical signal. Then we have the chemical signal, which is right here, and that is a molecule secreted by a cell used to communicate with other cells or the target cell, and that is the cell that will be receiving this chemical signal. Typically how it works is a cell secretes a chemical signal and attaches to the unique receptor on the surface of the target cell right here, and this alters the function of the target cell. So there are 4 different types of chemical signaling that we will go over, and that is paracrine, autocrine, endocrine, and direct signaling. So in paracrine signaling, we will just have a single cell releasing signals that will be sent to cells in the general vicinity of the original cell. And then we have autocrine signaling, which involves only one cell. Endocrine signaling, which involves a cell sending out signals through the bloodstream to cells all over the body. And then we have direct signaling, which involves cells that are right next to one another. Paracrine is going to be the first type of signaling that we're going to go over. So this is when a signal molecule is released by a single cell into an area and is received by multiple cells in that same area. This type of signaling is used in cases of stimulating cells in an area to grow and multiply. So an important example of paracrine signaling is in the development of the spinal cord. And so different levels of a certain signaling molecule called sonic hedgehog, or SHH, is released in the embryo while the fetus is developing. A gradient is formed as SHH diffuses away from our notochord, and the different concentrations of the molecule will tell which cell, which type of neuron it will become. So we can't talk about paracrine signaling without talking about synaptic signaling. And so synaptic signaling is very important in the nervous system because this is how our nerve cells are able to talk to one another. When an electrical signal passes all the way down a neuron, the signal becomes chemical, and these chemical signals are called neurotransmitters, so these little red dots. And these neurotransmitters are released from one nerve cell and diffuses across the space between the nerve cells, and this is called the synapse. And then it binds to proteins on the next nerve cell. This causes the signal to travel along the nerve cell from one nerve cell to the next. Next is autocrine signaling. And so this type of signaling only involves one cell, and a cell will release a chemical signal that will then bind to a receptor protein on its own surface, and it will trigger a response in the cell. So this kind of signaling is important, especially for cancer cells, because it helps for the cancer to metastasize or spread from one part of the body to the next. Next, we'll talk about endocrine signaling, and so this is the long-distance form of signaling. So another system called the circulatory system is utilized to help carry the signaling molecule from one cell to multiple cells all over the body. These molecules are made by specialized cells that will dump the ligands produced into the bloodstream, and these will be delivered to the appropriate cell. So you can see that these little dots, also known as hormones, will be dumped into the bloodstream, and they will go along until they reach a target cell. So, for example, is the hormone called human growth hormone, or HGH, and this is produced in the brain in the pituitary gland, specifically the anterior pituitary gland, and it travels all the way through the bloodstream to the muscles, to the bone and the liver and cartilage, and so by binding to all these cells, it stimulates them to divide, leading to overall growth in the body. Lastly, we'll look at direct signaling, and so all animals and plants contain cell junctions, which are little gaps in the membranes of the cell. So in animal cells, they're called gap junctions, and then in plants, they're plasmodesmata. And so these connect the cytoplasm of cells directly next to one another. So dissolved signaling molecules move from one cell to the next by cytoplasm. So, direct contact signaling is also involved in cell to cell recognition, and this is crucial in our immune response because if a cell called a macrophage engulfs a harmful invader, so like this bacterium right here, this is also called an antigen, it will present a piece of the invader on its surface, like these little orange triangles. And then this makes it so T cells can detect that there are harmful invaders in the body because the T cell has a receptor on its surface that will bind to the presented piece of the antigen. So right here, it's just detecting that there are antigens in the body. And so this T cell will then be activated, and an immune response will be activated as well. So now we will go over generally what happens once a signaling molecule comes in contact with a protein on the surface of a cell. So the first step is reception. And so the ligand attaches to a protein, like this one right here, on the surface of the cell. And most times the ligands are too large to pass through the cell membrane, while small ones can just pass through, like steroids. It is important to note that the molecule is specific to the receptor, so kind of like a lock and a key. This alters the shape of the receptor, in most cases, just activating it. So when talking about reception, it's important to go over the three major types of receptors that you can have. So the first one is the G protein receptor. And so this one has two parts to it, the receptor on the outside and then the G protein on the inside. So when a signaling molecule binds, the protein dissociates, like in the third part right here. So we've had the signaling molecule bind, and then the G protein has dissociated and it goes to an enzyme on the also on the cell membrane. And this activates the enzyme, and this triggers the signal transduction pathway. So the next is the receptor tyrosine kinase. And so this type of receptor is crucial in triggering multiple responses, and it can trigger actually up to 10 responses all at once by transferring a phosphate group from ATP onto the protein on the receptor, and this triggers a response. The last type of receptor we're going to talk about is the ion channel receptor. And so this is a type of membrane receptor that works like a gate, allowing molecules into the cell. And the ions that it will allow through are specific to the type of ion channel. And so what happens is the signaling molecule binds to the receptor, and this opens the gate, allowing the specific ions through, like sodium or calcium. And so when the ligand detaches from the gate, then it will close. So next in the process will be the transduction of the signal. And so what happens is once the signal has binded to the protein on the surface, the protein changes its shape as a series of changes are triggered in the cell. And so the signal is passed along a signal transduction pathway through relay molecules. And so you can either have proteins or you can have non-proteins that are small and can quickly move throughout the cell. And so how a signal is amplified is through this process. So a small number of signaling molecules can lead to a larger effect because more molecules can be activated. This also shows that the pathway creates as specific a response as possible. So a chain process occurs as one protein activates next and then next until the desired outcome is achieved. So lastly, we're just going to talk about the response part of the process. And so this response is due to the fact that the receptors are unique to the ligand. So as you can see in the beginning, that the signaling molecule is unique to the receptor. And so this triggers a very specific response. So once the signal transduction pathway is complete, the cell changes its activity. And so this is probably the most important part because this is at the end what is going to be your end product. So this could either be the turning of genes on or off, or regulating the production of proteins and or necessary molecules, and etc. So these are the topics that we went over, and I hope that helped.